In this video, we're going to continue to look at DNA replication in prokaryotes, and we're going to start with asking, or answering the question rather, how does Pol3 have such high processivity? Since Pol3 is the main prokaryotic DNA polymerase, we don't want it to fall off, especially from the leading strand. It needs to fall off from the lagging strand though, so how do we balance those two? And the answer comes in the form of this beta subunit or beta clamp. So this beta clamp will form a ring around DNA. And Pol3 will be attached to this. And that helps Pol3 not to fall off because it's attached to this clamp that's clamped onto the DNA. Now this clamp can move, it'll slide, it's called a sliding clamp. And that also helps speed up the rate of which nucleotides are added onto the DNA. Now the core, you can see here we have DNA going through it, but it's not a strong association, which makes a lot of sense. We're moving this clamp along the DNA, so we don't want it to stick to the DNA, just kind of encircle the DNA so we don't fall off. Now let's take a closer look at the different proteins that make up the replisome, the machinery that's actually replicating DNA for us in E. coli. And it's quite fast. We're doing about 1,000 nucleotides per second in E. coli. So let's take a look at this complex. So here in the green, we have our two different polymerase 3s. Remember, there's two in E. coli, one for the leading strand, one for the lagging strand. The yellow circle, that's your beta clamp, which the polymerase is hanging onto to allow it to stay onto the DNA. This green here is your primer, so you can see in this picture we are currently finishing up an Okazaki fragment. Our polymerases are connected through the C terminus of this clamp loader. So the polymerases are actually connected via this clamp loader protein. And also connected to this clamp loader, we have our helicase, DNA B, and our primase, G. So you can see right here, the primase has just added a new RNA primer, and the helicase will continue to move on to unzip DNA. And the DNA that's already unzipped has our single-stranded binding protein. So nothing happens to that DNA. It doesn't associate with anything. Now, what happens is that this clamp loader, this is what is loading the beta clamp onto our DNA, once for our leading strand and then multiple times for our lagging strand. Once the beta clamp's on there, Pol3 will come and bind to that clamp tightly, and that's how we get replication. For the lagging strand, every time we run into a previously synthesized Okazaki fragment, Pol3 will release so it can be loaded onto another beta clamp. But the beta clamp will stay there because that is what's going to recruit Pol1 to remove this RNA primer, and it also recruits DNA ligase to seal up the final nick for us. Once DNA, Pol1, and ligase has done their job, then the beta clamp will be released so it can be used on another Okazaki fragment. Now let's look at the actual reaction mechanism for a ligation reaction, or to seal up a nick and create a new phospho backbone for DNA. And we need energy to do this because we're making a new bond. So it's going to be through the hydrolysis of NAD plus to NMN plus AMP, or some organisms actually just use ATP for this. It depends on the species. For E. coli, we're going to use NAD plus. So lysine is conserved in all ligases. It's a critical critical amino acid for this mechanism to happen. So what happens in E. coli is first we have NAD plus, and we take the adenylyl group from NAD plus and put it onto the terminal nitrogen of lysine. And now we have this adenylyl group on our lysine. The second step is we just take this group that we just took from NAD and we stick it onto the 5' phosphotermini to form an adenylated DNA. Then ligase will help to catalyze this attack by this hydroxyl onto the phosphate, the 5' phosphate, kicks off our now AMP, and now we have a new phosphate backbone. However, this does not work with RNA. This DNA ligase will only work with DNA and DNA, which is good. You really don't hardly ever want RNA and DNA ligated together. So by having that 2' hydroxyl, you're going to make sure that ligase doesn't actually accidentally seal RNA into your DNA template. Now let's zoom out of the mechanism and look at the genome of E. coli in a whole. And what we find is that there's 10 nearly identical sites in the genome that are not palindromes. And they're called termination sites or Ter sites. So here's the OREC, the origin. Remember, from the origin, we're going to be replicating DNA in both directions. And the way to read this is, let's say that we are going counterclockwise and we're replicating. When we hit a Ter site that is facing towards us, we will stop. So if we're going counterclockwise, we're going to stop at Ter A. If we're going clockwise, the first termini we can stop is Ter C. Now, we will only stop if the TUS protein is there. So what happens is that the TUS protein will bind at these different Ter sites, and DNA B won't be able to remove that, and so our whole replisome will stop at that Ter site. However, this is kind of like a safety mechanism because we're replicating in both directions anyways, and as soon as our two replisomes run into each other, we're going to stop. They will collide and they will both fall off. This system, though, is strongly conserved, even though it seems redundant. So there must be some growth advantage we're not aware of that makes these TERs advantageous. Because you can kind of think of it as like a backup system, or like it's going to force the replisome to stop, but they would stop anyway. So we're not quite sure right now what is this growth advantage, but it must be there if it's seen in all different types of organisms. Or prokaryotic organisms, that is. Now, when our replisomes collide and we're synthesizing new DNA, they will accidentally become covalently linked and kind of crossed over each other. This is called a catenated chromosome, which isn't good. The black and green is one copy of a chromosome, the blue and red is the other copy. And we're trying to make a replication of our genome, but right now they're intertwined. And so we need to unwind this mess, this catenated chromosome, if we want to make a new organism, a new copy of ourselves. So the protein that's going to do this is called topoisomerase 4. And topoisomerase 4 will come, unwind this, so our chromosomes are now separated from each other. And with all the mechanisms we talked about, the replication of DNA is extremely precise. It has high fidelity, which means we don't make mistakes often. With everything combined, you only make one mistake every 100 million to 10 billion base pairs. So it's very rare that you make mistakes. They do happen, but it's very rare. And the reason why we have high accuracy is because we have balanced levels of DNTP, so 25% ATP, CTP, GTP, TTP. And so we're not going to just accidentally add the wrong base because it's overrepresented in our pool. So that's one way. Two, the polymerase reaction is a two-step reaction. One, we have the base pairing with the template that forms the hydrogen bonds we talked about. And once the hydrogen bonds are formed, then we can close to actually seal it in. But we're not going to make that seal unless we have the proper Watson-Crick base pairs. Three, we have 3' to 5' exonuclease activity. So that's supposed to be, say, function there. I don't know how that error happened. So if we do have an error, we're just going to go and snip it out and then we'll be ready to go and replace that error. And also, we have a whole other suite of enzymes that if this machinery does not pick up an error, it will go back and proofread again. And any error that happens will fix that. So a bunch of different proteins in each cell with the sole purpose of making sure our DNA is replicated correctly, which is smart because our DNA makes our cells who they are. So we want to make sure that the thing that's given directions isn't copied incorrectly. So throughout this, we've always said, we need an RNA primer to start. And what is the advantage of having an RNA primer? Why not just put DNA there, and so you don't have to go through this whole process of cutting out the primer and resealing it back in? And one of the reasons is that the first few editions of a primer are going to be super error prone. So the idea is if you put RNA there, they're going to chop off anyways. It doesn't matter how error prone your primer is. It's just a platform to get the polymerase to start. You're going to remove it anyways. So make all the errors you want. That's only temporary, and it's going to be replaced with what's correct later on. And with that, that's the end of this video, and I will see you in the next one.